సార్ లాస్ట్ క్లాస్ ఏమన్నా రికార్డెడ్ క్లాస్ ఉందా సార్?
లాస్ట్ ఇయర్స్ క్లాస్ ఉందని పెడతాను.
ఓకే సార్, థ్యాంక్ యూ సార్.
ఓకే. Good evening, students.
గుడ్ ఈవినింగ్ సార్.
Good evening, sir.
Today I'm going to explain you the Mendel's experimental design, various techniques followed by Mendel, and Mendel's laws, and a critical review of Mendel's laws. Okay. So coming to Mendel, we know that he is basically a teacher, or rather, he is a monk. Mendel is a monk. He has done certain teaching also in the monastery present in Austria, Austrian city of Brunn. But he has also done what we call it as research. But this research is not the research that is actually we do in laboratories. And this research is done in his monastery, in the fields, and basically this is called as a hybridization experiment. Okay. Mendel has a knowledge in mathematics, statistics, natural science, culture of fruits, that is pomiculture, and grapes, viticulture. In addition to this, he studied damage by a pea weevil called as Bruchus piscis, which attacked the garden pea. And this has made him to get interest in the study of peas. So why he has selected Pisum sativum for his hybridization experiments is that he has done a prior research before this on the same plant. And after getting Pisum sativum, what he did is he conducted certain hybridization experiments on the garden pea, scientifically called as Pisum sativum, for about 8 to 9 years, between 1856 and 1864. This is the period where Charles Darwin also is doing research on evolution. And Mendel is a contemporary of Charles Darwin. Okay? But they never met each other. At the same time, Mendel's research could not come into light because of certain things which we are going to discuss later in the coming slides. After all the research that he has done, in 1866, he published his findings with the title of Experiments on Plant Hybridization. And what he did is, he published his findings in the Society of Brunn, and the publication or the journal of the Society of Brunn is named as Annual Proceedings of the Natural History. This is the mistake done by Mendel. Why? Because these are the annual proceedings which are not circulated out of the Austrian country until the rediscovery in 1900. That means for about 34 years, his research is or was in dark, only because of the fact that he did not publish in a reputed journal which is what we call circulated worldwide. Okay. Is Mendel the only one who did research on heredity and inheritance? No. There are certain pre-Mendelian scientists who have done certain research. The foremost among them are Knight and Gauss. Why I am naming these two scientists is they were the pioneers, the first scientists in hybridization experiments on Pisum sativum. That means before Mendel only, there are two scientists who worked on Pisum sativum, but Mendel could succeed in formulating certain or giving certain conclusions, but the others, including Knight and Gauss, failed. We'll see why is it so. Coming to the experiment of Knight and Gauss. Knight's experiment was done in 1799. After 25 years, Gauss does the same on Pisum sativum in 1824. And after these two, that means in 1856, that means after 32 years of these two scientists, Mendel does his experiment. In Knight's experiment as well as Gauss' experiment, they have taken, one has taken flowers, flower color, pigmented and unpigmented, and they have got in the F1 generation, the first generation, the pigmented variety. When it was selfed, because Pisum sativum is a bisexual plant, when it was selfed, they got pigmented and unpigmented variety in certain ratio, which they did not mention. Okay. Let's say even they have mentioned the 3:1 ratio, but they could not do how this affected the entire process of heredity. Okay. And in the same way, Gauss also has done experiment on Pisum sativum with respect to the seed color. They have crossed the green seeded variety with the yellow seeded variety and got yellow seeds like Mendel got. They self it again as Mendel did, and they get again green and yellow. But what they did not do is they did not come up with certain conclusions. Why is it so? Let's see. The other scientists are similar observations were made by certain other scientists like Gartner, Norden, and Darwin in various other periods. In 1763, a scientist called as Kohlreuter, he crossed the tall and dwarf varieties of tobacco plant and found that all the first generation hybrids may resemble one or the other parent or maybe intermediate between them. That means either it is tall or it is dwarf or it is of intermediate height. This is the observation made by Kohlreuter in 1763 itself. But he did not make any conclusions. And at the same time, Gauss and Kohlreuter also discussed that reciprocal crosses gave similar results, which also Mendel does, but they failed to formulate any law by their experiments. Why the Mendel's predecessor failed? The first thing is the lack of numerical treatment of data. If you yourself or myself, anyone does research, what actually we do is we go for the statistical analysis of the data. How many plants were crossed? How many plants were born in the F1 generation? Of these, how many were self and how many gave rise to the F2 generation? In F2 generation, how many are said to be tall? How many are said to be dwarf? These are the numerical treatment calculations that have to be made so that you can give some conclusions. But Mendel's predecessors failed in numerical treatment of data. Why? Because their more importance is on improvement of varieties, producing high yielding varieties or crossing different colors and of flowers, producing another new color of flower, like that only, rather than understanding the mechanism of inheritance. The second thing the mistake that is done by them is they considered many characters at a time. Mendel has taken tall plant and crossed with a dwarf plant. But this the same tall plant is also having axial flowers, and this one may be having terminal flowers. The same plant is having flowers which are purple in color, and these are said to be white in color. The same plant is giving rise to seeds which are said to be yellowish and green. That means there are so many characters in the plant, and the Mendel's predecessor considered all characters at the same time. That is reason why maybe here this is dominant and this is recessive, this is recessive and this is dominant, this is recessive, this is dominant and this is recessive. Because they have considered all the characters at a time, they could not get similar results. But what Mendel does is he neglected all the other characters. He has taken only the stem length character in these individuals that made him a great researcher and successful. And coming to the Mendel's experimental design. So first one, we have to see the selection of the experimental plant. What he selected and why he selected. For example, if I want to do research, what I have to select is, because I am a zoologist, I have to select an animal. So what kind of animal I should select? If I select tortoise for my study, I want to study the life history of tortoise. Can I complete in my lifetime? No, because maximum I may live for 60 years. Already 25 years are gone. I am left with only 35 years. Whereas tortoise lives for 300 years, isn't it? So selection of animal or plant is the first success in your research. And now, after that, what you have to do? You have to devise a methodology of experiments. Mendel has devised certain methodologies. The first thing is he has selected all pure lines. All pure lines. What are pure lines? Pure lines are true breeding varieties. Okay. Second one, he employed emasculation technique. E means removal, masculine means male. So what is male here? In flower, anthers are male. The males anthers represent the male reproductive system. So he removed the anthers. The technique of which is called emasculation technique. And why he has performed, we'll see. And the third one is the most important, hybridization experiment. And he has done different kinds of process. One is monohybrid cross, where he considered only one pair of contrasting traits. Another one is dihybrid cross. He considered here two pairs of contrasting traits. And a reciprocal cross, back cross, and test cross. Based on this, Mendel made certain conclusions, which became laws afterwards, laws of heredity. We can also call them as principles of inheritance. Coming to the selection of experimental plant. So if you want to do any hybridization experiment, a suitable plant should have the following characteristics. One, it should have more characters to study. And the lifespan should be very short so that you can study more generations. In order to obtain true breeding variety, that means pure line, there must be a possibility for selfing. And hence Mendel selected Pisum sativum because it is a bisexual plant, selfing can be done, self-pollination. And the process of hybridization should be easier. So these are the certain criteria for selection of an experimental plant. So Mendel selected Pisum sativum for his experiments. Why? Because first thing is it has 7 pairs of contrasting characters. In the previous slide, we have seen that a plant for study should have more characters to study. So here it has more than 2 characters. That means it has 7 pairs of contrasting traits. And all these are easily distinguishable. That means they can be easily distinguished one from the other. You can easily distinguish a purple flower from a white flower, but not a dark purple from almost somewhat or light purple from intermediate purple. We cannot distinguish between. But these two can be easily distinguished. That is the reason why he has selected it. And the second one is he has selected Pisum sativum because it is an annual plant. Why? Because it completes its life cycle in 1 year. Okay, less than 1 year. And because of this fact, more generations can be studied within a short time. And the third character is bisexual flower. So this is a bisexual plant with which we can obtain pure lines upon self-pollination or self-fertilization. So these are the main characteristics by which Mendel has selected Pisum sativum. So let's see the 7 pairs of contrasting characters. The first one is tall stem and dwarf stem. Okay. And we call this character as stem length. So the first characteristic is stem length, which has two traits. One is tall, the other is dwarf. The second one is cotyledons. Some are said to be yellow in color, some are said to be green in color. Okay, color of the cotyledon is the second characteristic. And the third one is shape of the seed. Certain seeds are round shaped and certain seeds are wrinkle shaped. Okay. And the fourth characteristic is flower color. Certain flowers are said to be purple and certain plants are showing flowers which are white in color. And fifth one is pod shape, pod color, sorry. Yellow pod or green pod. And sixth one is inflated pod, which appears to us like a swelled one or a constricted pod. Okay. And last one, the flower position. Flowers are said to be axial or they may be at the tip of the branches, that is terminal. So these are the 7 characteristics studied by Mendel. First one is stem length. Second one is cotyledon color. Third one, seed shape. Fourth one, flower color. Fifth one, pod color. Sixth one, pod shape. And last one, flower position. So these are the 7 characters studied by Mendel. And let's see the first one, that is selection of pure lines in his experimental methodology. So how a pure line is obtained? So before that, let me tell you one thing. Why pure lines have to be selected? For example, there is a boy. Okay. This boy is white. Can you say that by just seeing this boy, his parents, father and mother, both are white? Can we say that? No. Why?
No, sir.
Father is white, mother is black, and still the child can be white. If father is white and mother is black, the child is white, but inside he has genes for black color. Thus, he becomes a hybrid one. For hybridization experiments, you cannot consider a character which is hybrid. What you have to do, you have to take a character which is a pure line. That means there is a boy whose skin color is white. His parents are also white. His grandparents are also white. His great grandparents are also white. That means for generations together, the skin color is same. That means there is no black color in any of these individuals inside in their genetic makeup. In that case, we can call them as pure lines. So when you want to select a white boy for to get married to a black girl, this one should be having only genes for white, both from his parents. So that is called as a pure line. And Mendel selected pure lines. How? A pure line is the progeny. Progeny means offsprings or children of a single self-fertilized plant. So there is this plant which is self-fertilized. It gave rise to progeny. Again self-fertilized. It gave rise to progeny. Again self-fertilized. It gave rise to progeny. And all the descendants of successive generations, if they exhibit the same character as seen in the previous generations, then these children are called as pure lines. Okay. So what Mendel does is these progeny exhibit the same character in successive generations because of self-fertilization of the parent plant. For example, a pure dwarf plant upon selfing will give only dwarf plants. Likewise, a pure tall plant will give rise only to tall plants. Thus, during hybridization experiments, inheritance of a particular character, that means whether it is tall or dwarf, it can be studied without any ambiguity, because this tall is not having any dwarf gene inside. It is not a hybrid. It is a pure line, so that there is no ambiguity and hybridization experiment can be studied easily. Okay. And what does Mendel do? He has taken a tall plant. He has self-date, and self. For example, he has taken a tall plant. This tall plant gave rise to tall plants as well as dwarf plant. So he removed the dwarf plant from the experiment. He has taken this tall plant, and this tall plant is again self, and it gave rise to tall and dwarf again. Let us say so. He removed the tall. He has taken this tall. He removed the dwarf and taken this tall. He again self. It gave rise to tall only. Again he has checked for another generation. Gave rise to tall only. It is again self and checked for another generation. And he can now conclude that this tall plant of this 1, 2, 3, 4, fifth generation is a pure tall variety because it is not having any dwarf character inside. In such a case, this is called as a pure tall plant. Likewise, a pure dwarf plant also can be produced. Okay, are you following?
Yes, sir.
Good. So the second one is emasculation technique. Why emasculation technique has to be employed? We all know that pollination can be of many types. It can be wind pollination, okay, bud pollination, insect pollination, anything. So here there is a flower. This flower is having the male reproductive system, that is androecium, represented by the stamens having anthers. So these anthers produce pollen grains. And the same flower is also having female reproductive system, that is gynoecium, represented by stigma, style, and ovary, isn't it? Now what happens actually in general, in nature? Here, this one, the pollen grains of this flower from the anthers fall on the stigma of the same flower so that self-pollination takes place. Okay. But what Mendel want to do, if this is a tall plant, what is going to happen here? Because of self-pollination, the same pollen grains of the same plant are going to fertilize the ovum of the same plant. But he want to do a hybridization experiment where he want to cross this with a dwarf plant, where this dwarf plant has again what we call bisexual flower. And this one also, what happens here also, self-pollination may take place. That means it may give rise to dwarf progeny only, this may give rise to tall progeny. But he wanted to know what happens if a tall plant is crossed with a dwarf plant. So the pollen grains of this plant have to be crossed with the ova, have to be fertilized with the ova of this plant. And for this reason, he is employing what we call emasculation technique. So what is emasculation technique? Let us see. So Mendel performed emasculation technique. It is used to perform, why it is used, why it is done? It is used to perform cross-pollination in a bisexual plant to produce hybrids. This is what you have to write down. So why emasculation technique is done? It is generally not done in a unisexual individual. Why? Because the unisexual individual has having its own male reproductive system. That means this is a male plant and there is a female plant. It is having its own female reproductive system. That means the pollen grains of this plant can be crossed with this plant. So there is no ambiguity here. But here, if it is a bisexual plant, both the reproductive systems are present in the same individual, then cross-pollination is not possible, because at any point of time, what happens? The pollen grains of the same plant from the same anthers in the same flower may fall onto the stigma of the gynoecium of the same flower of the same plant, so that there would be any hybrid at all. So what Mendel want to do? Mendel want to get a hybrid individual. That is the reason why he is performing emasculation technique. So what happens in emasculation technique? In emasculation technique, what Mendel does is he removed the anthers. He removed the anthers. So here there is a question. So there is a tall plant here. There is a tall plant here. There is a dwarf plant here. Okay. So this one is also having a flower and this one is also having a flower. And both are having androecium and both are having gynoecium. Okay. My question is, Mendel has performed emasculation technique. In which plant Mendel performed emasculation technique? Tall plant or dwarf plant or both? Okay. Mendel performed emasculation technique. My question is, Mendel performed emasculation technique in dash.
Dwarf
In this
plant.
present experiment, tall plant, dwarf plant, both, none of the above.
Dwarf plant, sir.
Dwarf plant,
Dwarf plants, sir.
only dwarf.
Both plants.
Both,
Both
sir. Yeah,
Yes,
both, sir.
plant.
Both. Mendel wants to stop the self-pollination. That's why he is used emasculation technique. I think it's maybe both, sir.
Somebody saying both, somebody saying dwarf.
సార్ ఏ
Which one is
ప్లాంట్ లో
correct?
అయితే స్టిగ్మా తీసుకున్నాడో ఆ ప్లాంట్స్ మాత్రమే అందులో మాత్రమే ఎమాస్కులేషన్ చేస్తారు సార్, అంటే యాంథర్ రిమూవ్ చేస్తారు.
Hmm.
I think he want to stop the self-pollination. That's why maybe maybe removal of both plants, or maybe.
Very good. See this definition. Emasculation technique is the removal of anthers from the flowers of a bisexual plant, which is considered as female plant. There are two plants, both are bisexual, and you cannot call one as male plant and the other as female plant because both are bisexual. But Mendel want to consider this tall plant as male, this dwarf plant as female. How he considered this tall plant as male? Because he has collected the pollen grains of this plant. Okay. And he collected the pollen grains of this plant and he dropped the pollen grains on the stigma of this plant. So here the ovary is present, the pollen grains germinate and fertilization takes place in this plant. So this plant, here what happens? Fertilization takes place between the pollen grains of tall plant, the ova of the dwarf plant. And because the ova are fertilized in the dwarf plant is considered as female. Because pollen grains are collected from the tall plant, here the tall plant is considered as male. Okay. But if he want to consider tall one as male and dwarf one as female, then what he has to do? He has to collect pollen grains of this plant and drop them on the stigma of the gynoecium of the tall plant. Then what happens? That the dwarf plant now becomes the male plant and the tall plant now becomes the female plant. So this can be either way. So what actually he's doing? He's considering one plant as male, the other plant as female. And some of you are saying that removal of anthers can be done in both, but that is not necessary. It can be done in both, but that is not necessary. Let us see. This plant is having anthers, okay? This plant is also having gynoecium in the flower. What happens? The pollen grains of this plant, this flower may fall on the stigma here. So in order to arrest it, what Mendel has to do? He has to remove the anthers. From which plant? From the plant which is considered as female. From the bisexual plant which is considered as female, so that cross-pollination will not take place here. Let us see this plant. So this is also having flowers. So there are anthers and there is gynoecium. And the pollination may take place here also. But here, this plant, the ova, the fertilized ova of this plant are not taken for what we call growing the next generation plant. The fertilization, the fertilized zygote or seeds of this plant, female plant, are said to be considered for growing the next generation. So here, whether it is fertilized or not, that is immaterial. What is taken here? Here you are taking only the pollen grains. You can just take the pollen grains from a paintbrush and paste them or place them here on the stigma. So here in this plant, whether anthers are removed or not removed, that is not a criteria because you are collecting only the pollen grains. Pollination has taken place or not, that is immaterial. Because you are collecting only pollen grains, here anthers need not be removed. Because you are fertilizing the ovum of the dwarf plant here with the pollen grains of the tall plant, here in order to arrest self-pollination from this female plant or from this bisexual plant, which is going to be considered as female, anthers have to be removed. So thus, we can define this like this. How? It is the removal of anthers from the flowers of a bisexual plant, which is considered as female, but not from the plant from which pollen grains are taken. Is it clear for everyone?
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
Okay. So so in which plant it is done?
Female plant.
Dwarf, sir.
It's not a female plant. It's not a female plant. It is the
It's a male plant.
bisexual plant. It is a
Bisexual
bisexual
plant.
plant which is considered as
Female.
That's what you have to answer. If anybody asks why emasculation in which plant emasculation technique is done, you should not say female plant because it is actually it's not a female plant. It's a bisexual plant. This bisexual plant is considered as female. Why it is considered as female? Because the ova of this plant are fertilized from the pollen grains of the other plant. Okay. So here in this first cross, the dwarf is considered as female and the tall is considered as male, and anthers are removed from dwarf. If you want to consider tall as female, what you have to do? If you want to consider tall as female.
Emasculate the tall plants. And
Very good, excellent. Emasculation has to be done in the tall plant so that it will become female and dwarf will become male. So you should never say he has done emasculation technique in tall plant, he has done emasculation in dwarf plant, he has done emasculation in female plant, he has done emasculation in male plant. These are the not correct answers. The correct answer is the emasculation technique is done in that bisexual plant, which is considered as female plant. Okay. So he has taken the bisexual plant. He removed the anthers. So only what is left here? Gynoecium left here. He has taken another bisexual plant. He did not remove anything. He just taken a painted brush, and that brush is said to be having the pollen, and that pollen are transferred onto the stigma of this female plant, this bisexual plant, which is considered now as female. So because pollen grains are collected from here, this becomes male. Because ova are fertilized here, this becomes the female. And seeds of only this plant, which is fertilized, only are sown to get the next generation. The seeds of this plant are not sown to get the next generation. Clear?
Yes, sir. I think it's not necessary to removal of another plant. I mean, both plants.
Yes.
Yes, sir. I have got it.
You have to remove only in this plant, the plant which is going to be considered as female. In that plant only you have to remove anthers. Clear?
Yes, sir.
Good.
Yes, sir.
Next technique is monohybrid cross. So I need not explain you about this. You already have listened to it more time. So I will have a quick run over. The cross between two plants differing in a pair of contrasting traits. What are these contrasting traits? Tall and dwarf. So one pair of contrasting traits of a single character. What is that single character? Stem length. Okay. So the cross between two plants differing in a pair of contrasting traits of a single character is called as monohybrid cross. So because here only one character is taken into consideration, this is called as monohybrid cross. Okay. Cross between tall and dwarf plants, cross between purple and white flower plant, cross between yellow seeded variety and green seeded variety, and so on. So what actually Mendel has done? So he has taken a tall plant, and this tall plant is pure tall plant. He crossed with a pure dwarf plant. And let us say this is male and this is female. Anthers are removed from the dwarf plant. And in the next generation, he could obtain all the tall plants. And Mendel has got a doubt at this point. Why the dwarf plant did not appear in F1 generation? So that is the reason why he has selected Pisum sativum. Because if there is any dwarf character passed from the parents to the offspring, the dwarf character should lie within the organism. If it is a unisexual organism, you cannot extract this out because self-fertilization is not possible. He has selected Pisum sativum so that it can be self, and whatever the character that is present, if at all inside, can be brought outside. So that is the reason why he has taken the F1 plants, all of which are tall plants, and he has performed selfing. He selfed them. So upon self-pollination, what he could observe in F2 generation is he could observe 3 tall plants and 1 dwarf plant. So the first question that came into mind here upon this first cross is, where has this dwarf gone? Okay. So in order to know, he has selfed the F1 plants. Selfing of F1 plants is said to be done. And what he could observe is the answer he could get. He could get the answer that they are present, they are found in the F2 generation. That means it did not go anywhere. It is present within the F1 individual only. That is the first answer that he has got. And he has got a second question. What is the second question? Here, this plant he knows it is a pure tall plant. And now this plant he knew after this experiment, second cross, that because it gave rise to tall and dwarf, This F1 individual is a hybrid. So thus, in plants, in these two plants here, one is pure tall, the other is hybrid tall. Okay. Now Mendel got the second question. What is the second question here? In the F2 generation, there are three plants. Please don't say there are three plants. There are 300 plants to 100 plants in tall and dwarf. In 300 is to 100 ratio. When we cross them, we can get 3:1 ratio. So now he wanted to know, of these 300 plants, how many are pure tall and how many are hybrid tall? And he wanted
100
to also,
tall,
he also wanted to
200
know,
hybrid tall, sir.
very good. He also wanted to know why this, whether this tall is said to be pure or not. So now he has gone for the, he continued the experiment till the third generation. Before Mendel, there are scientists, Knight and Gauss. They have done their experiments up to F2 generation and stopped there. And because they have stopped their experiments there, they did not know which of these three is said to be pure tall, that means homozygous tall, now we call them as, and which of these tall is said to be hybrid tall or heterozygous tall. So he continued his experiments till third generation and selfed all the 300 tall plants and 100 dwarf plants. And he could found that, let's say these are 4 in number. So 1/4 of the tall plants gave rise only to tall plants. Then we can say that these plants are pure tall because they gave rise only to tall plants. And all the dwarf plants gave rise only to dwarf plants, 1/4 of the generation. And they are said to be called as pure dwarf plants like the parents. So these two are pure lines like their parents. But the remaining 200, remaining two, they gave rise to, when this is self, it gave rise to 3:1 ratio, tall is tall into dwarf. And this one also gave rise to tall and dwarf in 3:1 ratio. Thus, these two are considered as, what they are considered as? Hybrid tall. How many are they? 2/4. And he got the ratio 1:2:1. So when you conduct experiments up to F2, then you get only 3:1 ratio. When you continue the experiments till F3, you can get the 1:2:1 ratio. Okay. And my question here is, okay, what is the result or the effect? What is the effect of continuing his experiments till third generation? What results Mendel has got by continuing experiments till third generation? Can anybody tell me?
He came to know the pure tall and pure hybrid and what are pure
tall ఒక pure dwarf
Very good.
ఇంకా రెండు hybrid అని వచ్చింది సార్.
So he could know which one is pure tall and which one is hybrid tall, isn't it? That is the conclusion, isn't it?
Yes, sir.
Which generation? F1 generation plants or F2 generation plants?
in F2 generation plants.
F2 generation plants. And we call this as genotype. Okay. So if any question in examination comes like this, by continuing his experiments till third generation, what results Mendel has got? Then you have to tell that Mendel got the genotype of the F2 plants by continuing his experiments till third generation. Please write down this. Okay. while all others stopped at F2 generation only. Mendel continued his experiments till third generation, and he got what? He got the genotype of the F2 individuals. One is pure tall, two are hybrid tall, and one is pure dwarf. That is what Mendel got. And we call them as now as homozygous tall, heterozygous tall, homozygous dwarf. And another conclusion made by Mendel in this experiment is, out of a pair of contrasting traits of a single character, only one character appears in the F1 generation. The character which appears in the F1 generation is called as, that means here this character is tall, is called by him as dominant character. The character of parental generation which does not appear in the F1 generation is found to be suppressed by the dominant character. Hence, this is called by him as recessive character. And now we can call this as homozygous dominant 1, heterozygous dominant 2, homozygous recessive 1. So thus, we have got two ratios, 3:1 and 1:2:1. In 3:1, we tell about whether it is tall or dwarf just. Thus, it is called as a phenotypic ratio. Whereas, in the F2 generation, we say about their genotype. Hence, N:1 is called as genotypic ratio. So in genotypic ratio, we represent this as like this. 1:2:1. 1 homozygous dominant, 2 heterozygous dominant, 1 homozygous recessive. You can simply write it as pure dominant, hybrid dominant, and pure recessive. So these are the things that we can learn from performing monohybrid cross. Okay. Mendel continued his experiments with all the 7 pairs of contrasting traits, and observed that there is a dominant and recessive relationship. Okay. And Mendelian dominance is called as a complete dominance. You have to write it down. Okay. Mendelian dominance is called as complete dominance. Why? When tall is crossed with dwarf, he did not get, let us say this is tall and this is dwarf. He could get only tall one, neither dwarf nor any intermediate one. When a purple variety is crossed with a white variety, and all the flowers in the next generation are purple only. They are neither pink or intermediate purple, or light purple, or white, dark white, light white, nothing like that. One is said to be completely expressing itself, and such a dominance is called as complete dominance. And Mendelian dominance is always a complete dominance. And the results of the 7 pairs of contrasting characters are like this. You already know this. We'll have a quick view of this one. First character that considered is stem length. The dominant is tall, recessive is dwarf. And the ratio is 3. These are the actual results Mendel that got. 2.84 means it's almost near to 3. So he gave us the 3:1 ratio. And there is 3.01:1. So it's also 3:1 ratio. 2.95, this is also rounded, and it gave rise to 3:1 ratio only. And he has considered 7 characters, and in all these 7 characters, stem length is having tall dominant and dwarf recessive. Seed shape, round is dominant, wrinkled is recessive. Seed coat color, yellow is dominant. We can also call it as cotyledon color. It is yellow is dominant, green is recessive. For flower color, purple is dominant, white is recessive. Flower position, axial is dominant, terminal is recessive. Pod shape, inflated is dominant, constricted is recessive. And for pod color, green is dominant, yellow is recessive. And how we have to represent? We have to take the English name of the tall plant. For example, for tall, the English name is, for this character, the English name is capital tall, and you consider tall factor as capital T and dwarf character as small t. And for seed shape, the first letter of round, capital R, small r. And for seed coat color, capital Y, small y, capital P, small p, capital A, small a, capital I, small I, capital G, small g. So this is how we have to represent different genotypes. And Mendel considered the second type of cross, that is dihybrid cross. So what is dihybrid cross? This is the cross between two plants differing in two pairs of contrasting characters. That means round seeds, wrinkle seeds, yellow seeds, and green seeds. So this one is said to be seed shape, and this one is said to be seed color. So 1, 2. How many? Two different characters are considered. Because two different characters are considered, this is called as di means two, dihybrid cross. So the individual that is produced will be a hybrid of two characters. Hence, this is a dihybrid cross. And characters are only two, but the contrasting traits are two pairs. So the cross between two individuals differing in two pairs of contrasting traits of two different characters is called as dihybrid cross. And example is this one. Cross between two plants differing in two characters, seed shape and seed color. Seed shape is round and wrinkled, and seed color is said to be yellow and green. This is dominant, this is dominant, this is recessive, and this is recessive. Okay. So why Mendel want to do this experiment? Why? Because he has observed that there are round seeds. Okay. There are round seeds, and he found that all the round seeds are yellow in color. All the round seeds are yellow in color. And he observed that all the green seeds are wrinkled in shape. All the green seeds are wrinkled in shape. So Mendel's doubt is why the yellow seeds are not wrinkled, why the green seeds are not round. Okay. What generally he observed is the yellow seeds are round, wrinkled seeds are green. Why not the round seeds are green? Why not the wrinkled seeds are yellow? So what he has to do? He has to make a hybridization experiment. So he has done, Mendel crossed a plant with round and yellow seeds with another plant having wrinkled and green seeds. All the first generation plants showed round and yellow seeds only, because round and yellow are dominant characters over wrinkled and green character. So if in F1 generation we got only round and yellow seeds, and he has self-pollinated. He wanted to see where this has gone. So the only thing that he can do is, because it is a bisexual plant, he can self-pollinate it. When self-pollination is done, okay, he observed that both the parental characters are appeared. That means round and yellow is there. Wrinkled and green is also there. That means these are called as parental characters in next generation. In addition to this, he also got new combination. That means round instead of yellow, it give rise to round and green seeds. Wrinkled instead of general green, it gave rise to wrinkled and yellow seeds. And these are called as non-parental combinations. So in the hybrid cross, he has got two types of plants. One is called as parental combination. The other is called as non-parental combination. So what is the difference between these two? Parental combination is the one which is present in the parental generation. Round and yellow, wrinkled and green. Non-parental combinations are new combinations. We also call them as recombinants. They are new combinants or recombinants. What are they? round and green, wrinkled and yellow. So thus we get two types of what we call individuals. In what ratio? 9:3:3:1 ratio. Of these, the 9 are parental combinations and 1 are parental combinations. The central ones are non-parental combinations. So when Mendel performed his experiment, if it is a Mendelian dihybrid cross, you have to remember both of these, those that are present on the extreme right and extreme left, are said to be parental combinations. Those that are present at the center are non-parental combinations. So how many non-parental combinations are there? 6. How many parental combinations are there? 10. So total 16 are said to be produced. Okay. And he wanted to know the genotype of these plants. So what he does what we call the self-pollination technique again, and he got the next generation individuals, and he got the genotype of the individuals. So how we'll represent the genotypes of these individuals? I'll give you a small, a brief picture so that you can identify the genotypes easily. So let us say capital R, capital R, capital Y, capital Y. This is the parental generation plant, round and yellow, small r, small r, small y, small y. So what happens here? Capital R to one, capital Y, capital R to the another gamete. So capital Y, this one. Likewise, small r, small y, small r, small y. So this is the parents and these are the gametes of the parents. So when these are fertilized, capital R, small r, capital Y, small y. And these are also round and yellow. And this is F1 generation. Now he has taken the F1 generation individual and he crossed it, self-pollinated it. So this is the androecium of the bisexual plant. And this is the gynoecium of the bisexual plant, not two plants, same plant, selfing is done. So now what happens is crossing over, now what happens is 4 different types of gametes are produced. How we have to represent? First you separate capital R from small r and combine with capital Y. This is one gamete. And again, capital R, take capital R, combine with small y. So two combinations of capital R is done. Now do the same with the small r. Take small r 2 times, combine with capital Y once, combine with small y once. Thus, we get 4 types of gametes. This will be asked in question in examinations. How many types of gametes are produced from an F1 hybrid in a dihybrid cross? How many types? 4 types. Whereas in a parental generation, how many types of gametes are produced here? In the parent generation, how many types of gametes are produced? Here?
2 types.
Who is going to tell? 2 types or 1 types? 2 types or 1 type?
2 types.
Capital R, capital Y, both are only one type. So in parental generation, so what you have to write is, in parental generation, you get only 1 type of gamete. In parental generation, all gametes are same. That means it is only 1 type of gamete, capital R, capital Y or capital R, capital Y. And if the parent is said to be small r, small y, it is either small r, small y or small r, small y. Only 1 type of gamete is produced. Whereas from the F1 hybrid, in a dihybrid cross, you get 4 types of gametes. So according to Punnett square, we go for a checkerboard. So we take all the gametes, male gametes on one side, female gametes on the other side. So gametes are always circled. Don't forget. So capital R, capital Y, capital R, small r, capital Y, small r, small y. Likewise, capital R, capital Y, capital R, small r, capital Y, small r, small y. These are the gametes. So you know better than me. You can simply capital R, capital Y. Likewise, capital R, capital R, capital Y, small y, and so on. So how to identify among these? If blanks are given, genotypes are given, and if someone asks which one is said to be round and yellow, which one is said to be green and wrinkled, we can simply see from this one. Okay. When you see this one, so this diagonal or this triangle, how many? 1, 2, 3, 4, 5, 6, 7, 8, 9. All these are round and yellow. Okay. So this diagonal is round and yellow. Next, second one. What is the second one? What you have to get? Parental combination is wrinkled and green. So what is the wrinkled and green? Only one, the last one. So this is the last one, wrinkled and green. So this is only one, 9:1. Then coming to the other two. What are they? Round and green, wrinkled and yellow. Round and green, wrinkled and yellow. So where round and green are present? So for wrinkled and yellow and round and green. So for wrinkled and yellow, what you have to consider is you have to consider these three, this triangle. So this triangle is wrinkled and green, wrinkled and green, and wrinkled and green. So these are three. So we are left with only three boxes. What are they? This one, and this one. These three are round and green. These are wrinkled and yellow, and these are, wrinkled and yellow. These are round and green. So thus, from this checkerboard, you can easily see what the different types of genotypes are, and phenotypes are. So thus, in a dihybrid cross, we get how many phenotypes? We get 9:3:3:1. This is one phenotype, second phenotype, third phenotype, fourth phenotype. We got only 4 phenotypes. How many? 4 phenotypes. Then coming to the genotypic ratio. So the genotypic ratio is like this: 1:2. Again, 12 2s are 24. 2:4. Again same, 1:2. Again repeat, 1:2 and 1. This is the ratio which you have to by heart, or at least you can remember it like this: 12 followed by 12 2s are 24. Again 12, again 12, again 1. 1:2, 2:2, 2:4:1:2, 1:1:2:1. This is the genotypic ratio. So how many genotypes are present here? For dihybrid ratio, how many phenotypes are present? 1, 2, 3, 4. 4 phenotypes are present. Then how many genotypes are present now? 1, 2, 3, 4, 5, 6, 7, 8, 9. So we get 9 genotypes. So how we have to represent it? So let us see monohybrid cross and a dihybrid cross. The phenotypic ratio is 3:1, 9:3:1. And genotypic ratio is 1:2:1. And here it is 1:2:4:1:2:1:1. Okay. And now what we have to do is we have to see the number of phenotypes and genotypes. So there is a small formula. So number of phenotypes here, how many are there? 2. Here, how many phenotypes are there? 4. Okay. That means the formula for phenotypes is 2 to the power of n. 2 to the power of n. So here n refers to the characters under consideration. In a monohybrid cross, only one is considered: stem length. So 2 to the power of 1, the number of phenotypes are 2. And coming to the hybrid, how many? 2 characters are considered: seed shape and seed coat color. So 2 square. This is 2 square. Thus, the number of phenotypes are said to be 4. And coming to the number of genotypes. How many genotypes are there? 1, 2, 3. How many genotypes are here? 1, 2, 3, 4, 5, 6, 7, 8, 9. And the formula becomes 3 to the power of n. That means here it is monohybrid crossroad. So that 3 to the power of 1, that is equal to 3 only. Here dihybrid cross, 3 square. 3 square means 3 into 3, that is equal to 9. So this is how we have to write down. And my question is, there is a dihybrid cross ratio. I'm going to tell you the phenotypic ratio. 27 followed by 3 9s followed by 3 3s. 9 3s are 27. Okay. And 1. This is the phenotypic ratio. Genotypic ratio it's very big. So in no examination, trihybrid cross or for tetrahybrid cross, genotypic ratio will be asked. But they may ask the number of phenotypes and number of genotypes. So who is going to tell me the number of phenotypes and number of genotypes for trihybrid cross based on the formula given above? Did you understand my question? Hello?
8 phenotypes, sir.
The phenotypes are 8. Why?
2 to the power of 3, sir.
3. Excellent. 2 to the power of 3. Here n refers to 3 because it is a trihybrid cross. Then what about the genotypes? Number of genotypes? Number of genotypes? How many?
27,
27 will be the number. So based on this formula, you can easily say for monohybrid, dihybrid, trihybrid, tetrahybrid, if any cross is given, you can say the number of phenotypes and genotypes easily by the formula 2 to the power of n, 3 to the power of n. Here it is 2 followed by 2 square 4, 2 square 4 followed by 2 cube 8 followed by 2 to the power of 4. How much? 16 followed by 32. Likewise here 3, 9, 27, then 81, 243 and so on. Okay, please note this down. Please note this down. Okay, did you complete?
Yes, sir.
Okay. So now we are coming to the end of this topic, experimental design. So that is reciprocal cross. The third one is reciprocal cross. Why Mendel performed reciprocal crosses? Because he wanted to test his results. Mendel tested his results with reciprocal crosses. So what actually happened is, he has taken a tall plant, he crossed with a dwarf plant. This is considered as male and this is considered as female. That means pollen grains of this plant is considered and this is crossed with or fertilized with the ova of the dwarf plant. Then came a question into Mendel's mind. What happens if I consider the pollen grains of this plant and ova of this plant? So the dwarf becomes the male and tall becomes the female. So what actually happened here is, in the first cross, tall is crossed with dwarf. In the second also, tall is crossed with dwarf. That means in both these two crosses, the characters are kept the same. What is changed? In the first cross, tall is male and the same is female in the second cross. In the first cross, dwarf is female and which is male in the second cross. That means here in this, these are two crosses where the characters are same, but the sexes are reversed. And such a crosses are called as reciprocal crosses. And Mendel wanted to know, is there any influence on the inheritance of these characters, the influence of sex on the inheritance of these characters? So when Mendel crossed in the first cross, the results are said to be tall. A tall plant crossed with a dwarf plant where the tall is said to be male, dwarf is said to be female, the results are said to be all tall plants. Okay. When he crossed a tall plant with a dwarf plant, with the tall plant being female and the dwarf plant being male, the results are same, again tall. So based on this, he can conclude that there is no influence of sex. There is no influence of sex on the 7 pairs of contrasting characters Mendel has taken into consideration for his hybridization experiments. Why? Because all these are somatic characters. Because all these are somatic characters. Okay. So in a reciprocal cross, the characters involved in the cross remain the same. And what is reversed? The sexes are reversed. Okay. You write this down. In a reciprocal cross, the characters involved in the cross remain the same. Only the sexes are reversed. Okay? Completed?
Yes.
Good. And the next thing you have to mention is results of reciprocal crosses are similar. Results of reciprocal crosses are similar. So these are the 2 points which we have to remember when somebody asks you anything about a reciprocal cross. So a reciprocal crosses are not just one. Reciprocal crosses are 2 crosses. In the first cross, in the second cross. In both the crosses, characters are same, but only the sexes are reversed. And the result is said to be same. Why? Because all the characters considered by Mendel are somatic characters. And here also there is a question for you for tomorrow. Mendel is saying that results of reciprocal crosses are similar. My question is it a universal phenomenon or not? Okay. So by the time you come for tomorrow's class, tell me whether reciprocal crosses are same. Whether the statement of Mendel that reciprocal crosses are same, is it true or false for all the different living organisms present on the face of the earth? Next. So this is the first cross where the tall plant is said to be male and dwarf plant is said to be female. In a reciprocal cross, the characters remain the same and only the sexes. This became female and this became male. And the result is said to be tall only. Thus, the results of reciprocal crosses with respect to Mendelian inheritance are same. And the last third one is said to be back cross. So what is a back cross? Back cross is a cross of a hybrid. That means F1 individual, capital T in this case, with any one of the parents. So what are the parents? Capital T, when it is crossed with small t, it gave rise to capital T, small t. So these are the parental plants. This is the F1 plant. And in this cross, this is the F1 hybrid, which is crossed. That means capital T, small t, crossed with capital T, capital T, small t, crossed with small t. That means it can be crossed with any of the two, either homozygous dominant or homozygous recessive parent. Then That cross is called as a back cross. Why we actually perform back crosses? It is a process of getting what we call it's a technique employed in hybridization process in both animal breeding as well as plant breeding in horticulture. In addition to this, it is also responsible production of gene knockout organisms. Okay, and backcross is performed. If somebody asks you why backcross is performed, backcross is performed to produce new varieties in plant breeding as well as animal breeding. And second one is to produce gene knockout organisms. So what gene knockout organisms are, we'll discuss later. So in a monohybrid cross, when the F1 hybrid is crossed with a homozygous dominant, all are said to be tall plants. The same F1 hybrid, when crossed with a recessive parent, homozygous recessive parent, we get tall and dwarf in 1:1 ratio. And this back cross result, this one is our test cross. Okay. This one is our test cross. And the ratio of test cross is always 1:1 with respect to monohybrid cross. Why we'll see now. So test cross. So let us see what test cross is. I'm asking you one question. I have given you a tall plant. I have given you another tall plant. And I am asking you to tell me which one of this is pure tall. That means homozygous dominant. Which one of this is hybrid tall, heterozygous dominant? Can you say which one is pure tall or hybrid tall just by seeing the plants? Can anyone say? No. Then how you are going to tell the genotype of these two plants?
By crossing them.
What happens if when they are crossed? By seeing what kind of result you're going to say this plant is pure tall plant. So this is the biggest question. So Mendel wanted to know why test cross is done. If anyone asks you why test cross is done, what you have to tell is pure and hybrid means genotype. Test cross is employed to know the genotype of the given dominant plant, not the recessive plant. Because recessive plants genotype is always smaller only. When somebody gives you dwarf plant and asks you its genotype, you need not go for any hybridization experiment. You can simply say that it is smaller. Because no dwarf plant can be capital R smaller because there is complete dominance, capital R suppresses a smaller gene. So here, the recessive plant is always having the same genotype. You need not go for a test cross. Why you have to do a test cross? You have to employ a test cross only to know the genotype of the dominant. This you have to tell anyone. So if somebody asks you why the test cross is done and in which plant, test cross is done to know the genotype of which plant? The dominant plant. In other words, test cross is done to say whether given a dominant plant is homozygous or heterozygous. This is what you have to say. So test cross is a cross between an organism with a dominant phenotype. So you want to know the genotype of this one, the tall plant. So what actually you do? You cross it with a genotype of known individual. So what is the known individual, known genotype? Recessive one. This is the. So when you want to know something of one, you have to refer it to the one which is said to be known. So this is the unknown genotype. This is the known genotype. So in test cross, what you do? You cross the unknown genotype, dominant one, with a known genotype. So the test cross is a cross between an organism with a dominant phenotype, which is a known genotype, unknown genotype, whose genotype is unknown, and an organism that is homozygous recessive for that genotype, whose genotype is known. So here you are crossing two individuals. One individual with unknown genotype, the other individual with known genotype. Known genotype is always recessive. Unknown genotype is always dominant because it can be homozygous or heterozygous. Whereas known genotype recessive is always homozygous. So what happens in this cross? So when you cross a tall plant, this tall plant can be either this one or this one, capital T or small t. You are crossing with this one with a dwarf plant, that's the small t. So let's see the first case. So you cross the capital T with small t. What happens? These two, anyone, it gives rise to only capital T small t. That means all plants are tall. Let us say, take the second one, capital T small t into small t. So it produces two kinds of gametes. Here it produces one kind of gamete, capital T small t combines to form capital T small t. And here small t. One is tall, the other is said to be dwarf. Thus we can get 1:1 ratio of tall and dwarf plants. Okay. So if it is tall, the given plant is said to be tall, all when it is crossed with a dwarf plant, all are said to be tall plants only. Then you can say that the genotype is said to be homozygous. When this is said to be heterozygous, tall, all the plants will be that are produced will be 1:1 ratio, tall and dwarf. So just by, without having any representation of capital T small t, you just have taken a tall plant and you crossed it with a dwarf plant and you got all tall plants. You just tell me what is the genotype of this one. Okay. Are you following?
Yes, sir. I think it's a heterozygous or homozygous.
I'm not telling you anything. I'm not telling you anything. The result I am telling. The result is that all are tall plants. So let's see. Small t, capital T. Then only all are said to be tall. That means it is homozygous. In another cross, I have given a tall plant and we crossed it with a dwarf plant. And you got tall plants as well as dwarf plants in 1:1 ratio. How is it possible? The genotype of this one is not going to change. And you can get only a tall plant when this tall plant is said to be having a small t gene. That means it is a heterozygous individual. So just by seeing the result, you can say that the genotype of the dominant plant. So thus, in first cross, when somebody says a tall plant is crossed with a dwarf plant and the resulting plants are all tall, okay, then you can say that this is a pure tall plant. In the second cross, a tall plant is crossed with a dwarf plant and you got tall plants as well as dwarf plants in 1:1 ratio. Then you say that this tall plant is a heterozygous individual. Just by performing this experiment without going for the genes, you're not mentioning anything about the genes here. You're just seeing the result of the cross. Just by seeing the result, you can say the genotype. And thus we are testing whether the dominant one is homozygous or recessive, homozygous or heterozygous. That is the reason why this type of test cross is said to be called as test cross. Okay. So here, when all tall plants are said to be produced, the given plant is homozygous. When tall and dwarf are produced in 1:1 ratio, the given plant is said to be heterozygous tall. So this is the test cross ratio for a monohybrid cross. For a dihybrid cross, the test cross ratio will be the phenotypic ratio of monohybrid cross is 3:1 and test cross is 1:1. That means there are two phenotypes, we get two phenotypes only. For dihybrid cross ratio, 9:3:3:1 is the phenotypic ratio. And the test cross ratio will be, here there are four phenotypes, we also get four phenotypes, 1:1:1:1. If it is a trihybrid cross, the phenotypic ratio is 9:7, 27:3:9:3:3:1. How many? 8. That means here the genotype will be having 1:1:1:1:1:8 ones will be there. That is how you have to mention the genotypes. So for monohybrid cross, the phenotypic ratio and test cross ratio. Phenotypic ratio is 3:1, test cross ratio is 1:1. For dihybrid cross, phenotypic ratio is 9:3:3:1. So that the test cross ratio will be 1:1:1:1. So what you have to remember is, how many varieties are there here? 4. So here how many ones will be there? 4. Here how many phenotypes are there? 2. So how many ones will be there? 2. And for trihybrid cross, the phenotypic ratio is like this. How many? 1, 2, 3, 4, 5, 6, 7, 8. 8 phenotypes are present. So the trihybrid cross ratio will be, 1:1:1:1:1:1:1:1:1. 8 ones will be there. This is how you have to represent the trihybrid cross, test cross ratio of monohybrid, dihybrid and trihybrid crosses. Is it clear?
Yes.
Okay, write down. If you have any doubts, ask me. Otherwise, we'll close this class today and we'll discuss about what we call Mendel's laws tomorrow. And their significance. Critical review. Don't miss tomorrow's class.
Okay, sir.
Any doubts, ask me. Otherwise, we'll close here.
Sir, previous class record పెట్టండి sir.
Okay, you already asked it. Elson.
Thank you, sir.
Shall we close now?
Yes, sir.
Yes, sir, you
No
can.
doubt, sir.
Yes, sir.
Okay,
Thank you,
sir.
sir. Thank you.